I don't think, because last semester nobody took one. And so I didn't write one. So I don't have one written. So I will do my best to get you one.
I can always just use the old chapter.
Yeah, you can do that, and you can also use the learning objectives from the big study guide. I will try to get one. To be perfectly honest, that will probably fall to the bottom of the pile because I have to write one for my 102 class because I don't have one for that class either.
Gotcha.
So,
okay.
and I know by the end I will be writing, that'll be 4 exams I have to write plus 101. 101, at least I don't have to write a new exam or a new cumulative, so that's good.
That's fair.
But I don't know what I would do if I wasn't teaching that class in addition to this in 102, because this one and 102 are always a lot of work. But that one I just kind of show up. And I'm like, oh, I've taught this like 16 times 2 sections every semester. So
Yeah.
that one I could probably do in my sleep. I literally like show up to that class like, what are we doing today? Perfect. Conveniently, in there we are also doing replication and transcription translation. So it is really nice.
Just review.
Yeah. Okay. So before we get started, do you all have any questions about what's going on in class or anything? Okay. So chapter 9 is all about DNA replication. On Wednesday last week, we went through the Meselson and Stahl experiment. We went really, really quickly, but the gist of the Meselson and Stahl experiment was that they showed that DNA replication happens by the process of semi-conservative replication. And in semi-conservative replication, each parent strand is used as a template to make the daughter strands. So we have our parent strand here. The gist of DNA replication is that the parent strands are going to pull apart. And then each new daughter strand is going to be built using the parent strand as a template, following the rules of complementary base pairing. And what you end up with are 2 pieces of DNA that are identical to one another. So today we're going to learn how that happens. We're going to learn about all the enzymes that are involved, and then we'll also look at what happens to the sequences when we're doing this, because we can kind of think about it in terms of the overall strand, and then we can also think about it in terms of the actual sequence. So just a couple of different terms. One of those is replicon. So a replicon is just a short fragment of replicated DNA. We're going to see in just a minute that our double strands have to open up, and then DNA polymerase works to build short fragments at a time at various replication starting points. So we're going to I probably won't use the term replicon very often. I'll probably use the term fragment, but if you hear replicon in your smart book or in your textbook, that's what they're talking about. In bacterial cells, we have something called theta replication. Theta replication is the replication that happens on a piece of circular DNA. I'll show you a picture in just a minute, but basically what happens is that at one point in your double-stranded piece of circular DNA, you get a little bubble that opens up, and then your replication enzymes bind to the starting point in that bubble and replicate going around and away from the start of replication. In eukaryotic chromosomes, where we have linear DNA, we're going to get multiple origins of replication. Multiple places where we get little replication bubbles that open up, and at each replication bubble, we're going to extend our replication from the origin out away from the origin. So when we talk about DNA replication, we're going to be talking about generally E. coli replication. We've mentioned several times that E. coli is a common model organism in genetics, and that's because it's really small, it's easy to grow, and a lot of the processes, even the molecular processes, are really, really similar. So in eukaryotes, they tend to be a little bit more complex, but the general idea is the same. And so we're actually going to be talking about E. coli replication, but it's applicable to linear replication as well. So this is a diagram of theta replication. So you can see we have a circular chromosome here. And in our bacterial chromosome and in our linear chromosomes, you have origin points called origin of replication. And that is just the place where the DNA is going to open up, and our DNA polymerase and our other replication factors are going to come in and actually start replication. Usually our origins are AT rich. So that doesn't mean there are no C's and G's. It just means there's a high concentration of A's and T's. Sometimes it means that you get a small segment that is only A and T. And this is because there are only 2 hydrogen bonds that connect adenine and thymine. So those 2 hydrogen bonds are easier to break and pull apart than the 3 hydrogen bonds that happen between guanine and cytosine. So it's just a little easier to open up the DNA strand there. The adenine, thymine rich regions are also going to be found at our origins of transcription. So when we get to gene expression in the next chapter, we're going to see these regions as well. These are called promoter regions in transcription, but they're called origins of replication in bacteria and in eukaryotic chromosomes. So what's going to happen at our origin of replication is we're going to get this little bubble that opens up, and it is called a replication bubble. So as we are talking about replication, we're going to kind of zoom in on what's happening from our origin of replication and moving away from our origin of replication, because replication is actually a little bit different depending on sort of which quadrant you happen to be in and focused on for replication. So we're going to talk about how replication differs depending on where it's actually happening within that origin of replication. For bacteria, we're going to start at that single replication bubble, work out and away all the way around the circle on each strand until we get both of our strands replicated. Then our bacteria do some fancy things where they make a little strand break here, separate the 2 pieces of DNA, and then they're actually pulled apart and partitioned into the new daughter cells. So remember, our goal for DNA replication is actually for cell division. So to copy our DNA and then partition it into the cells for bacteria into the clones, and then for mitosis and meiosis to the new daughter cell that's coming up. So we talked about replication happening before mitosis and meiosis, but we didn't actually talk about what was going on here. For linear replication, the idea is the same, except along our long linear chromosome, we're going to have multiple origins of replication. So up top, we've got our linear chromosome. We've got several origins along our chromosome. And in each one, they're going to open up, again, an AT-rich region there. We have a series of enzymes that are going to come in, pull apart that bubble, and then DNA polymerase is going to come in and start copying. In eukaryotes, when we have these multiple origins, they're basically going to sort of grow into one another. So bacterial chromosome, we have 1, it just replication extends out from that bubble. But in our linear replication, each one of these is just going to continue pushing open the replication bubble, and eventually those bubbles are essentially going to fuse, and we're going to end up with a single long replicated chromosome from each of our strands. So our in our eukaryotic cells, we're going to end up with fragments that are going to sort of merge into one another. Each of those fragments, or each one of those replicons, is 200 to 300,000 base pairs long, depending on how far apart those origins of replication are. So if you move along the chromosome and look at the sequences, you can see multiple origins of replication all the way along the whole linear chromosome. So one of the handouts I gave you, this one, is a list of all of the enzymes that are required for DNA replication, and then just a brief summary of what it does. This is for bacterial cells. Eukaryotic cells actually have a few more things, and they have multiple versions of DNA polymerase that have slightly different jobs in their cells. So that one is just the bacteria ones. Those ones are the ones that you're going to need to know, be able to identify what their function is. All right, so for replication to take place, we need our double-stranded DNA template. We need nucleotides, and we need some enzymes and other proteins that are going to help this process along. So floating around in the cells, in the nucleus, I should say, there are a bunch of free nucleotides, our adenine, guanine, cytosine, and thymine. So before we get into the actual process, just really quickly, I kind of wanted to show you what's happening with the sequence. I'm just going to make something up here. Remember that our DNA sequence is anti-parallel. Anti-parallel means that they run in opposite directions with respect to their backbones. So on the 5' end, actually, I'm not going to tell you. Can somebody tell me what we have at the 5' end of our DNA strand? Which part of our nucleotide do we have at the 5' end of the DNA strand? No. Put your hand.
The base.
Not the base.
Is it the primer?
Nope. We need a primer though.
Pure.
Oh.
Is it the hydroxyl?
Oh, all right. Amir is the closest. It's the phosphate, yes. So at the 5' end, we have our phosphate group. And then where do we find the hydroxyl, Amir?
3.
At the 3' end. Very good. So at the 3' end, thank you, Jacob. 3' end is the hydroxyl.
I had those two mixed up too. That's why I was.
The 3' end is the hydroxyl group. So the 3' end for DNA replication is actually going to be our important one. That 3' end is where DNA polymerase is going to come in. And where DNA polymerase is going to add our nucleotides. So we'll come back to this, but since we're talking about the ends, DNA polymerase only ever adds to an existing OH group. So DNA polymerase can't just randomly start adding nucleotides in the middle of a strand. It must have a 3' OH. And a minute ago, Lindsay mentioned primers. So primers are going to be required for that to happen. Okay, so just looking at what's going on with the sequence here, by way of a quick review, our sequences are anti-parallel, meaning one end has a free phosphate, the other end has a free hydroxyl group, then the opposite strand is runs the other way. So our strands are not the same. They are complementary. So if we read this forwards or backwards, they're going to be different. What that means is that each strand can actually encode different genes. So in what is the top strand in this image, this top strand could encode one gene. The bottom strand in another direction could encode another gene. They could be overlapping because the sequences are not exactly the same on both sides. They're complementary, but they are not the same. The complementary base pairing is important though, because if we know one side, we always know the other side. And we can use that complementary sequence for both replication, and when we get to chapter 10, we're going to use it for transcription. So semi-conservative replication says that DNA polymerase is going to use each parent strand as a template for the new daughter strand that we're building. So we are actually going to build our strand in a 5' to 3' direction. When you write these out on paper, you don't have to do that. I'm just going to do it just to illustrate the function of DNA polymerase. So DNA polymerase is going to come in, and it's actually going to build it starting with the 5' end and working its way towards the 3' end. So if we make the complement, that's what we would get. If we make the complement on the other strand, feel free to call me out if you see any mistakes. Hopefully I don't make any, but it's very likely I will. And so what you should end up with are two exactly identical pieces of DNA. One that is made up of the parent strand and the daughter strand, and the other one that is made up of the other parent strand, the other daughter strand. And actually, I should have probably drawn this one on the inside because they're facing one another, but it's not super important here. Yes, Lindsay. Did I make a mistake? Oh, which one?
The last G on the daughter strand.
Oh, I did. Look at that. Lindsay gets an extra credit point today. That's the only way I get my 101 students to answer questions. So if I tell them I get an extra credit point for catching me.
I wasn't going to say anything.
Well, now you're going to say it. So I will tell you that when we get to transcription and translation, you get extra credit points for calling me out on calling it the wrong name. And now that transcription's in my head from 101, I might even do it in replication. So now you're looking for mistakes. I can, like, a lot of times I will catch myself before students will catch me. So, but it does keep 101 students engaged. All right, so replication is the easy one. We're going to just keep all of our base pairs. Remember, DNA has thymine. Once we get to transcription, we're going to have RNA, and so we're going to have uracil instead of the thymine. But DNA is always going to be double-stranded. Parent is a template for the daughter strand. Okay, so replication is going to happen in opposite directions on opposite strands, because of DNA polymerase's quirky little function that it can only add to that 5' end. So if we have strand of DNA, 5', 3', let's assume that it's already opened up. Just like our last image, our DNA polymerase is going to come in. We're going to need that little primer that Lindsay mentioned. Oops, this is on the wrong strand. Or on the wrong end of this strand, sorry. We're going to need a little primer. We mentioned primers last week when we were talking about PCR. We'll go over PCR in a little bit more detail since we were racing to get out of here last week, which I assume you all got home okay since you're here today? Yeah. Okay. All right. So we are going to build our new strand. running in that 5' to 3' direction. Same thing here, 5' to 3'. So depending on where our origin of replication is, which strand and which direction our molecules are running, one of our strands is going to have what we call continuous replication. The other strand is going to be replicated in little fragments, and we call this one discontinuous. So we'll talk about what I mean by that in just a minute. But one of our strands is going to be made continuously from the origin of replication towards the replication fork. And then the other one is going to be replicated discontinuously, essentially from the replication fork not really towards the origin. From the replication fork is not really the best way to explain it, but kind of from the replication fork. Okay, I'm just going to put it in quotes because that's not really what I want to say, but that's what I'm going to say for now. And this one is going to be made in small fragments that are going to have to be connected together. These are not the same thing as replicons. The strand that's made in little fragments are made in really short fragments, not 200 to 300,000 base pairs. So these are going to be little fragments that are just being built sort of within the replication fork. Okay, so there's nothing really new on this slide except to show you at the molecular level what's happening, because we're not going to draw the structures when we're going through kind of drawing our big replication bubbles. So this diagram just illustrates what's happening on our DNA strand. So on this image, the gray one is the template. This is the parent strand. And then the sort of orangey tan color is the new strand. You'll notice we've got our sugar phosphate backbone here. And we've got our nitrogenous bases in the center. Adenine and thymine connected by two hydrogen bonds, cytosine and guanine by 3. So what's going to happen is we've got this free OH here. DNA polymerase is an enzyme and this enzyme is going to catalyze the formation of a bond between the 3' hydroxyl and the 5' phosphate of the next nucleotide. So we have in our DNA polymerase reaction a slightly modified set of nucleotides. So if you guys remember from Biology 101, we talked a lot about ATP. ATP is a modified adenine nucleotide. ATP stands for adenosine triphosphate. So this is an adenine base, but instead of having only one phosphate group, it actually has 3 phosphate groups. So our nucleotides that are being used during DNA replication are actually triphosphates. So they are nucleotide triphosphates. And the reason for this is they are acting as sort of their own energy source. So ATP in the cell during cellular reactions that require energy, ATP works by kicking off a phosphate group, and when it kicks off a phosphate group, that group is usually transferred to something else in the reaction. Now, when this happens, there's a transfer of energy, and that energy is used to perform whatever chemical reaction happens to be taking place. So our cells, rather than using a whole bunch of ATP, use these nucleotide triphosphates. And every time one is added, they basically bring in their own energy in the form of these phosphate groups and use that energy to catalyze the bond that happens between that OH group and the phosphate group. So those two phosphates are broken off. The energy is used instead to build that bond here between the OH and the phosphate. This forms a bond called a phosphodiester bond. And that's just the fancy name for a bond that is a phosphate to an oxygen. So we're going to bond this oxygen to that phosphate group. All right. Not too much to see here, except this is the replication fork that we are going to be talking about. So this is if we have a replication bubble that starts at the origin, and then each of these areas on the side are called a replication fork. So we are going to kind of look at our replication bubble and our replication fork at just one side. So you can kind of think about the replication bubble as having sort of 4 quadrants. And so we're going to look at half of the bubble. In this case, this image is kind of showing the right half of the bubble. And what I would like you to keep in mind is that on our replication bubble, the same thing is happening in the opposite quadrant. So we're going to talk about our continuous and discontinuous replication. On each strand, if we have continuous replication here, we are also going to have continuous replication on the opposite quadrant. So if we have continuous replication in region A, we're also going to have continuous replication in region D. The same is true for our fragments. So if we have fragments or discontinuous replication in region B, we are also going to have discontinuous replication in region C. So on both of our strands, we have both continuous replication and discontinuous replication. But overall, each strand is still being built 5' to 3'. So if we look at this strand up here, I know that it shows all just continuous. But if we look at this strand being built 5' to 3', this strand being built 5' to 3'. And then as our replication machinery moves further and further away from the origin of replication, it's going to push this replication fork open. So as it moves, it's going to push it wider and wider. Okay. So a couple of new terms, or more new terms. This is leading and lagging. So our leading strand is the strand that is undergoing continuous replication from the origin towards the replication fork. So if I go back to this image really quickly, I don't know why I drew it so tiny, but up on the top A, starting at the origin, moving towards the left-hand replication fork, A would be our leading strand for the top piece of DNA. Oops. And then D would be the leading strand for the bottom. I'll come back, don't worry. And then the opposite quadrants are what we call the lagging strand. So the lagging strand is the one that's going to be made discontinuously. So here, B and C are lagging strand regions. So these two strands are made just a little bit differently. Leading strand is going to be made continuous. Lagging is going to be made in these small little fragments. And then those little fragments are going to be connected together. All right, so those little tiny fragments are called Okazaki fragments. They have a fancy name because the guy that discovered them decided they should be named after him, or someone else decided they should be named after him. I'm not sure what, but so those little tiny fragments are on the lagging strand are called Okazaki fragments. In this image on the right, this one is showing the top strand being made in fragments. And the bottom strand on this one is the continuous one. So you can actually always figure out which one is going to be leading and lagging. So no matter which strand is on top, you can actually always figure out which quadrant you're in and which one is going to be leading or lagging. So again, that leading one is going to be built towards the replication fork. The lagging strand is going to be the one in fragments. And it's kind of generally being built moving away from the origin of replication. That's not 100% true, but to sort of describe what's happening there, it's the one that's kind of moving away from the origin. So, or from the fork. So if you think about this as the origin of replication, the bottom is our leading strand because we're being built in a continuous strand starting at the origin, moving towards the replication fork. The lagging strand, the other one, is actually starting in the replication fork and moving away. And as the bottom strand DNA polymerase pushes that open, we're going to get fragments built as it opens up. All right, before, do I want to do that yet? No, I don't want to do that yet. Nope, we went there anyway. Okay, before we actually get into DNA replication, I want to show you a little video of how replication, so you can kind of keep in mind what's happening. Our sound works in here now. I finally took care of whatever problem that was. All right, so I'm going to just go ahead and let you watch the video, watch what's going on. It's going to go through all of the enzymes that are going to come in, but kind of give you a big picture idea of what's happening. And actually, I just changed my mind about which video I want to show you. I'm going to show you a different one first. I'm going to show you a simpler one. All right. Hopefully we have sound. Fingers crossed. So if you kind of recall what was happening over here, you've got on the top strand, the DNA polymerase pushing the replication fork open. And over here, off of the screen, you've got a replication fork. So back here is the replication fork, and it is opened up and left the sort of unreplicated region. So that's what's happening here. Okay. All right. In a few minutes, I'm going to show you a different video that has more correct diagram. Oh, we're not learning DNA sequencing today. All right. Let me figure out where we were. All right. So DNA replication is going to start with what we call initiation, followed by a process of elongating the DNA strands. So the first thing we need are initiation proteins. These initiation proteins are going to kind of direct all of the other proteins into the DNA replication origin and DNA replication complex. So we're going to see some parallels when we get to transcription. We're going to have some initiator proteins and the initial proteins coming in, all to sort of guide the correct enzymes into the right place. So first thing, that initiator protein is going to bind at the origin of replication in the eukaryotic chromosome. This is called ORI-C for short, origin of replication. I'm not sure why the C, but so it's going to bind at that origin, and then our other important enzymes are going to come in. So there's a few things that we need. We need that initiator protein to sort of act as a signal or a signpost for the other enzymes to come in. We're going to need DNA helicase, which we'll come back to in just a minute. It's on your enzyme list. This is going to open up the start unwinding at the origin. We're going to need some proteins that are not enzymes, but these guys help keep the double strand apart, keep it open. These are called single-stranded binding proteins. And then we also need an enzyme either called DNA gyrase or DNA topoisomerase. I'll probably call it topoisomerase because that's just what I'm used to, but your textbook, I think, calls it gyrase. So we will talk about what all of these things are as we get there, but that's what we're going to need. Up there on the right, we've got an image of that initiator protein coming in, binding the DNA. And then when that happens, basically what it does is causes a negative supercoil. So we talked about DNA supercoiling and twisting up really tightly. You can also have a negative supercoil, which is where you unwind it the opposite direction, and that causes it to sort of open up the opposite from basically unwinding. And then DNA gyrase is going to come in and force apart those double strands. All right, so here's a more detailed image. So what we have is our origin of replication. So the replication bubble opens up at our origin. These little purple proteins, those are single-stranded binding proteins. If they were not there, those complementary base pairs would just want to close back up to one another because the hydrogen bonds make the nucleotides, the nitrogenous bases attracted to one another. So it just would want to close right back up on itself. Then we get DNA helicase at each of the replication forks. So this guy is kind of going to act like a zipper pull. If you think about a zipper on your jacket, it's going to sort of force open the replication fork. But we need one on both sides because we're going to be replicating in both directions. If we were to just open these, the DNA molecule could, if we were to just force open this replication fork, we could actually cause breaking on either side, because as you pull open the replication bubble, you're actually going to force the molecule to tighten its twist on either side. So the DNA molecule can't just freely rotate within the cell. It's bound up with histone proteins. It's bound up with other replication origins. If we're on a linear on a linear DNA. In bacteria, if we just open it up, we're just going to twist up the helix tighter on the other side. And so we get DNA gyrase that comes in. DNA gyrase actually sort of does controlled breaking. It allows the molecule to break at specific places and unwind the tension that's in that molecule. Because remember, we've got a circle. So if we just keep opening it up, we're going to twist it all up in other places. So topo means to twist. So what the term topoisomerase means an isomer is a different form of the same molecule. So topoisomerase allows the same molecule to twist into a different shape, is basically what it's doing. It is going to relieve some of the twisting stress on that molecule. I should have my rubber bands to show you what's happening here and kind of show you on these little ones. I don't have the big ones with me because I don't have my office with me. But you can kind of see what happens on little ones. If you twist up your DNA strand, twist up your rubber band into a double helix, and then you might need a partner depending on how easy it is to do it. But if you twist it up and you keep twisting it, it's gonna twist back on itself. That's supercoiling. If you do it right, if you can put your twist on like your index finger and your thumb, like this, and then don't make it too tight, but make a twist. And then if you go to try to open up the center, you watch as you open up the center into what is equivalent of a replication bubble, you'll notice that because the ends can't untwist, because in bacteria it's a circle, or in eukaryotic cells there's other origins there, you'll notice that that helix, that double helix gets really, really tight on either end. In a cell, it's not rubber, right? So in a cell, this would cause bonds to break, cause the molecule to break. So DNA gyrase's job is to come in, loosen up the twists on either end, and allow the molecule to rotate so that it does not break. So helicase is what's going to hold open at either end of that replication bubble at the forks. So, well, now I've lost it. So DNA gyrase, where the purple is going to kind of work to push open those forks. And then I've got my fingers holding the replication bubble open. That's how your single-stranded binding proteins are going to work. I'm going to kind of hold that open for you. All right, so process of replication during elongation. Again, DNA polymerase cannot add to that to anything but a 3' end. So what that means is that once we open up our replication bubble, DNA polymerase can't just come in and start adding nucleotides. We need to build a 3' end that it can work off of. So first, RNA polymerase is going to come in and start the replication. RNA polymerase is going to come in and make a little set of RNA nucleotides as a little primer. And then what that's going to do is leave a 3' end that DNA polymerase can work on. Now, there's two reasons that it's RNA primase. One, RNA primase does not need that primer. RNA polymerase can come in and just stick in a nucleotide at any point that it wants to. The reason that RNA polymerase can do this is probably related to what we call RNA world. We'll talk about RNA world when we get to chapter 10, but the RNA world hypothesis basically says that RNA was the first coding molecule. And so RNA replication probably evolved first when there were far less enzymes. And RNA polymerase does pretty much all of these jobs all in one. It opens up the strand, it starts the strand, it replicates the strand, making new RNA. And so the reason that it's RNA is probably twofold. One, because RNA polymerase can do that, can add without the 3' end. But the reason it does that is probably that it evolved first, and it evolved to do that in the absence of any other proteins. So that's why it's RNA, not DNA polymerase here. But we're trying to make DNA, and so we're going to have to replace that DNA with RNA. Sorry, RNA with DNA in a little bit. All right, let's go ahead and get down the definition of what the different components are doing, and then we will take a break and go through the process of DNA replication, and I will draw out what's happening for you. I think that all or most of these should be on your handout that I gave out, so you may not need to rewrite them all if you just want to refer to your handout, but they are there in your slides if you would like to do that.
Most of these are enzymes, right?
Mm-hmm. The only ones that are not technically enzymes are the initiator protein and the single-stranded binding protein. All the other ones are technically enzymes. Because the single-stranded binding proteins are not catalyzing any reaction. They're just sort of there blocking it going back together. And then the initiator protein is really just forcing that negative twist. It's not really like doing any reactions.
You know, like the structural difference between the 1 and 3?
Polymerase 1 and 3?
Yeah.
No. I do not. I have actually, so I do know, I know a little bit about the DNA polymerase 3. I know what that one structure looks like, but I don't know what the polymerase 1 structure looks like. And I'll talk about polymerase 3 in just a few minutes. All right, and actually, I'm going to go ahead. I'm going to go ahead and have you write down the general steps. The only thing that's not in the general steps is the difference between the leading and the lagging strand synthesis. So when we come back from break, I'm going to draw out what's happening and kind of work through what actually happens during elongation of both lagging and leading. So first we have to add in the primers, then DNA polymerase comes in. We have to remove the primers and then link all of our pieces together. All right, so I will leave this up for those of you that are writing this down. If you are all finished, go ahead and take a 10-minute break, and when we come back, we'll draw out what's happening during replication.
You good?
Okay.